my name is Surbhi Trivedi. I'm also from Harris Lab. My project this summer, and that I've been working on actually for quite a bit of time now, is assessing dengue virus intrahost intrahost diversity in human infections. And so, like I said, I'm from Harris Lab. I work with another undergraduate. Her name is Meghna, and I'm mentored by postdoc Dr. Purnima Parvaswaran. Okay. So the primary goal of my project is to profile the sequence diversity in viral genomes during acute infections with dengue virus in humans. So how do these mutations accumulate, and how does this diversity that I'm going to talk about accumulate in the host? So as you can see, this is the RNA viruses. Dengue is a single-stranded RNA positive sense virus. But the orange dot was the RNA replicase that the virus codes for itself. This is an RNA-dependent RNA polymerase, and as it's replicating, as it's creating new copies of the viral genome, it has no error-fixing mechanism, so it's rather error-prone. So it creates lower fidelity copies of these genomes. So you can see at a rate of about 1 nucleotide in every 10,000 nucleotides that it replicates, there is an error. So these mutations accumulate at various loci along the genome, and this is what we denote as the intrahost virome. So you can see this collection of different genomes with mutations along the genome. So a precedent for our study was in different viruses, including HIV and hepatitis C virus, where researchers found a correlation between the extent of intrahost diversity and disease severity. That's not exactly the question that we're asking, but it does set sort of the framework for studies such as ours. Okay, so a quick background. This has been covered by the previous presenters from my lab. There are 390 million cases of dengue infection per year, per the latest estimates, and 3 billion people are at risk in tropical and subtropical climates, according to the World Health Organization. And these climates where dengue is endemic because dengue is a mosquito-borne virus. So while 75% of infections are asymptomatic, 1/4 of these infections do exhibit clinical cases of these symptoms. The majority of these symptoms are this dengue fever, which is a febrile illness. It's debilitating but self-limiting, so generally not life-threatening. However, a fraction of these clinical infections do result in more severe cases of dengue hemorrhagic fever and dengue shock syndrome, which are life-threatening and characterized by vascular leakage. So one of the characteristics of dengue virus that has been covered by my presenters is that it exists as 4 different genetically related serotypes. And these serotypes have 60% conservation genetically between them. Where this dimension plays into is the disparity between primary and secondary infections. So a primary infection is simply the first infection of the virus of the individual, and the secondary infection is simply the second infection. If that second infection is by a different serotype, so a heterotypic infection, then those individuals that are infected are at a higher risk of developing these more severe cases, these more severe clinical cases of dengue hemorrhagic fever and dengue shock syndrome. And given the massive epidemiological burden of dengue virus, it's concerning that there's still no licensed vaccines or therapeutics for dengue. Okay, so in our study, we looked at individuals that were infected with dengue serotype 3 during the 2009 to 2010 serotype 3 epidemic in Nicaragua. All of our samples, the blood samples that were collected, were during the acute phase of the infection, so days 1 to 3 post-hospitalization. These were all patients with either primary or secondary dengue infection, and mostly clinical cases of dengue fever. So the samples that we worked with for processing purposes were both plasma samples and PBMC samples, so just 2 different types of samples that we have. The plasma is simply blood, leave that. So the plasma samples are simply blood minus the cellular component. So what's in plasma? It's circulating viral RNAs, which is so important. It's important for our study, as well as clotting factors and circulating proteins. The PBMCs are the white blood cells in the blood. So these are mainly the cells of the immune system, including B cells, T cells, monocytes, macrophages, and NK cells. So those are the 2 types of samples that we're going to process to answer our question. So we have these two types of samples, serum and PBMCs. First, what we did for our methods was to process them to extract all the viral RNA, extract that nucleic acid. Then we converted the RNA which is relatively unstable and harder to work with, to complementary DNA, which is easier to work with and better for sequencing downstream. We converted it to cDNA using reverse transcription. Then we amplified that cDNA using PCR. So PCR simply creates more copies of the DNA that was present, creates more sequences. Okay, so the way that this PCR was designed, it was designed with a specific intention in mind. So 90% of the genome that we ultimately covered was captured twice by the PCR products. And the purpose of this is to ensure that all the genome is covered, all the genome is represented in our sequencing data later on. So if a particular PCR maybe didn't amplify the DNA as effectively, then an adjacent PCR can still compensate for that region of the genome, and we can still ultimately process that with the sequencing data. This is just the genome of the dengue virus. Towards the 5' end, you have 3 structural proteins, the capsid, the premembrane, and the envelope proteins. And the rest of the genome codes for non-structural proteins, some with known functions, some with unknown functions. Specifically of interest is NS5, which is the viral replicase. So that's the polymerase that is replicating copies of the viral genome during the course of the infection in the infected individual. So ultimately, we successfully processed 63 PBMC samples and 42 serum samples, including these replicates, and the purpose of the replicates was to ensure reproducibility of our results and to weed out any false positives. Okay, so continuing for our methods, once these PCRs were amplified, what we did is we gel purified them using gel electrophoresis, and then we sequenced the purified PCRs. We ligated a unique nucleotide barcode to each of the samples. We pooled them so that we could very efficiently and rather quickly sequence them. And we sequenced them using this newer sequencing technology known as high-throughput sequencing, which allows for deep sequencing of the sample that you are trying to process. And this is on the order of millions of sequences, millions of reads of the sequence that is of interest. And per sample, we were able to generate hundreds of thousands of DNA sequences. And so the only way to be able to fully analyze this and properly analyze this huge data set is using bioinformatics. So my postdoc coded Python and Perl scripts to be able to, because it's like coding. I'm just gonna like, since I know it's like tiny, it's like all the slides are like a little too fast, so everyone's like, okay, right, so after generating all of the sequencing data, what we looked at, first we looked on the scope of the genes and domain level. So this is many nucleotides and analyzing where we might find higher rates of diversity on that scope. What we found, interestingly, was we found higher diversity in the envelope gene, and I'll talk more about what the implications and what the role of the envelope gene are in the dengue virus. So we found envelope gene had higher diversity compared to other genes, and specifically, the envelope gene has 3 different domains. Within it, envelope domain 3 showed higher diversity compared to the other domains. And also interestingly, which I'll talk about the implications of this result, envelope domain 3 showed differential diversity between primary and secondary infections. And there are implications to this as well. Okay. So this is a mature dengue virus structure. On the surface of virion, you can see the arrangement of the envelope protein. So this illustration shows 2 envelope proteins that are arranged in a head-to-tail homodimer. This is a native arrangement of 2 envelope proteins, and you can see it has 3 different domains to it. It has domain 1, 2, and 3. 1 is red, 2 is yellow, and 3 is blue. And where this red triangle is located, that is the AB loop of envelope domain 3, and that is where we found higher incidence of diversity compared to other regions of the dengue virus. So since the envelope protein is on the outer surface of the virus, it is one of the interfaces between the host's immune system and the virus, like the infecting pathogen. So antibodies have been identified that bind to all 3 envelope domains, and specifically the region where we found higher diversity, envelope domain 3, has the additional role of binding to cell receptors and facilitating viral entry into the cells. Okay, so after looking at the scope of many nucleotides, so the gene or domain scope, now we're zoning in and looking specifically at nucleotides, and a particular nucleotide we'll define as a locus. So our question now is, what particular locus exhibits higher variation and higher mutation, and are these variants also present in many samples? Okay, so this plot shows, so along the x-axis you can see the different locus, the different nucleotide across the viral genome, and the viral genome is 10,000 nucleotides long. So each of the points along the x-axis is a particular locus in the viral genome. And the red dots correlate to that locus where we found 80% of the individual samples showing higher viral mutation at this particular locus. So that's kind of significant. Specifically, the nucleotide mutation that we found was a directional nucleotide, so instead it was a transition mutation from an A to T. So instead of an adenosine, we found this thymine mutation to predominate, and we saw it was basically exclusive. We didn't see we saw very low rates of transversion mutations between A and C and A to G. Okay. So this region, this AV loop, where the mutation was found, is highly conserved. It's highly conserved not only in the dengue serotypes, but across the flavivirus genus. So this includes West Nile virus, which is located over there. This AV loop, in particular, what we're looking at is codes for the histidine amino acid. So the mutation location codes for a histidine amino acid. Okay, so the mutation that was found from an A to T change changes the histidine that was supposed to be coded for into a leucine mutation at that particular position in the envelope gene. And this led us to ask, what is the role of the histidine residue? Well, the histidine is located again in envelope domain 3, and the role of that region in particular is for viral fusion of the endosomal membrane. So what does this mean? So what happens when the virus is entering the cell? The virus binds to a cell receptor on the membrane of the cell. And then through receptor-mediated endocytosis, the cell phagocytizes and allows the cell to enter allows the virus to enter the cell. But in order for the virus to be able to proliferate and to infect the cell, it needs to gain access to the cell resources so that it can replicate copies of the cell and proliferate. And in order for this to happen, then the virus membrane needs to fuse with the endosome membrane, and that occurs and is facilitated by the particular region where we found the mutation. So that's kind of significant. Okay, so again, asking the question, why a mutation at this location? We have several hypotheses. Of course, we need to test them to be able to adequately understand what the implication of this mutation is. So first, we're going to assay the serum antibody binding and neutralization properties of the mutant virus versus that of the wild-type virus that lacks the mutation. And what we're going to do is engineer an infectious clone with this particular mutation in the virus and assess the serum antibody binding and neutralizing properties of that virus with that particular phenotype. And secondly, we're going to ask, how does this particular mutation affect viral fitness? So do the wild-type virus and mutant virus show any differential replicative abilities between them? And we're going to use this assay in cell culture, so in vitro. So in summary, the particular hotspot of diversity that we found is located in envelope domain 3. It shifted a histidine 315, that particular amino acid, to a leucine. We also saw higher incidence of variants in secondary infections as opposed to primary infections. And our hypothesis for this is that secondary infections have naturally a higher pre-existing antibodies that exert a particular selection pressure that might cause this mutation, this particular mutant virus, to be more prevalent. Of course, we need future experiments to be able to validate our hypothesis. And alongside that, we're going to continue sequencing the plasma samples that have already been processed And by this dimension of our project, we'll be able to assess the difference between intracellular and extracellular viral diversity and see if there's any bottleneck effect that shows a difference between the viral diversity within the cell and without the cell, so extracellularly. Okay. All right, thank you. I'd like to thank my project funders, of course, Surf Rose Hills, as well as my many mentors in the lab. This has been a very incredible experience, definitely. And okay, thank you for listening.